Okay, are we ready? Okay.
Yes.
Alrighty, hey guys, welcome to our presentation on bringing an innovative device to market. For this session, we'll be outlining regulatory and quality strategies for the novel bare metal coronary stenting system, also known as the NBMCSS, which we'll be referencing to that throughout. Our main focus will be on four key components. First off, the QMS flowchart and its specific interactions. We'll then go over the overall device evaluation strategy, which would include non clinical laboratory studies plans, and then also a clinical investigation plan. Ultimately, these elements will be used collectively to ensure compliance with FDA regulations and demonstrate safety for the effectiveness. Now, here's just another quick agenda visualizing the four main parts, and then we'll also finish with a conclusion section and key takeaways.
Before we discuss our quality system and testing plans, let's establish what the NBMCSS actually is. Our device consists of two main components, a balloon expandable bare metal stand and its delivery catheter system. The intended use is to treat patients with coronary artery disease by improving blood flow through narrowed vessels. Because it's a life-sustaining class 3 implant with no substantially equivalent Radicate device, we must pursue the PMA pathway rather than 510K clearance. This regulatory classification drives our entire development strategy, from the comprehensive non-clinical testing plan to the pivotal clinical trial design you will see throughout this presentation.
So diving into the quality management systems, we see that management oversees all of the subsystems. Everything comes from it and goes back to management. So with design controls, we define what the stent must do, including biocompatibility, strength, and fatigue life. These design requirements directly influence material selection, since only certain alloys and coatings can safely meet those requirements. At the same time, material properties will feed back into design by limiting achievable stent geometry or performance. With each material purchased, having serial and lot numbers help with identification of the material and will be recorded so that if something does go wrong, developers can document issues but also track down patients if there was a recall. All design and material decisions and changes are captured through document control, which ensures that only approved specifications, procedures, and records are used throughout manufacturing.
Howdy, this is Bailey, and this slide focuses on the operational components of the quality management system and how manufacturing equipment and corrective actions are controlled to ensure consistent device quality. As for equipment and facility controls, they ensure that the manufacturing occurs as a controlled and validated environment. This includes routine equipment calibration, preventative maintenance, and personnel training. These controls are critical because uncalibrated equipment or untrained operators can directly compromise product quality. As for production and process controls, this governs how the stent is actually manufactured. This includes validated manufacturing processes, sterilization controls, and final acceptance testing. For an implantable coronary stent, these controls ensure that each device released meets predefined specifications for safety and performance and can be overall repeatable in manufacturing. As for the CAPA, or the corrective and preventative action, it serves as a feedback mechanisms of the QMS complaints, deviations, and non-conformances triggers investigations through root cause analysis. Corrective actions address existing issues, while preventative actions reduce the likelihood of reoccurrence. Together, all of this ensures manufacturing issues are detected early and corrected before it impacts patient safety. So now we're going to look at our QMS flowchart and how all of these design controls interact with each other. Obviously, management responsibility is all of these controls. They allocate resources, conduct management reviews, and establish overall regulatory strategy, including planning. Design controls ensures that the stent is developed systematically from user needs through verification and validation. This includes defining intended use, translating requirements into design inputs and outputs, and verifying performance. Design outputs feed directly into material selection, equipment requirements, and production processes. As for material controls protect against variability introduced upstream. So that directly ties into production and process controls. As for equipment and facility controls, it supports production by ensuring that all manufacturing equipment is calibrated and maintained. This directly ties into design controls, which feeds into it, and then again, management's responsibility. As for production and process controls, materials and material controls feeds directly into this, and then it goes directly into records and change control, so we know what to change, and directly feeds into the CAPA requirements, so we can get that feedback and change as needed. And together, these interacting elements form a closed-loop quality management system that supports FDA compliance, readiness, and patient safety for the novel bare metal coronary stent system.
And for our final table, this just shows details about how the major quality subsystems interact with and influence one another. While they all influence each other in some way, design documents and CAPA will have a large influence on the development of the stent. In meeting pre-market approval, design specs are super important to ensure that the performance of the novel device meet the intended use. With any purchases of materials, results of process validation, or changes in the design process, everything is required to be documented. This documentation then informs CAPA, which will then dictate what changes may need to be made to any subsystem to ensure that the device adheres to all regulations in place, which will then allow the stent to be approved for consumer use.
Howdy, my name is Nicholas, and I'll be going over the device evaluation strategy. So first of all, the purpose of the device evaluation strategy is to ensure that the FDA understands the value of the information that we give them. This basically means that if we perform a test and give them the information, this tells them what it does and why we need it. This ensures that both us as well as the FDA have more efficient use of our time and efforts. So we went over through some of the some important topics regarding the testing. So one of them being the implant integrity, or the integrity of the stent. So some possible failures are the failure of the structure. This can cause a fracture in the actual stent, and this could cause trauma to adjacent structures nearby the stent, as well as embolization downstream from the stent. Information that we would need to reduce the risk include strength testing, as well as stress and strain analysis of the stent. We also wanted to look at the deliverability of the stent. This can happen if something like the balloon ruptures or the stent dislodges, or if there's any issue in actually delivering the stent to the target area. So some potential things that can happen is that the stent can be damaged, or you can even be unable to use the stent during the operation. Some possible effects are restenosis as well as thrombosis, and some information that we could use to better prevent this is the balloon burst pressure. Something else that we were also looking into is the biocompatibility of the stent. So this has many different failure modes, but one possible one is hypersensitivity response, and it can potentially cause thrombus formation in the stent area, and this thrombus can also lead to myocardial infarct or as well as inflammation. So to further reduce the risk of this, we need to look at the cytotoxicity as well as hemocompatibility. Next slide, please.
Howdy, this is Sahana, and I will be going over more of the device evaluation strategies to add to what Nicholas said. And so one of them is still stent expansion. So that happens when the stent doesn't expand symmetrically. And so the altered stress concentrations can actually lead to fractures, and this can essentially cause thrombosis and abnormal blood flow. And so what we would need to do to kind of support the testing is deployment tests at different pressures so that this doesn't affect the patient and lead to the thrombosis or abnormal blood flow. And another problem that could happen with the device is corrosion with the stent specifically. And so what would happen is this would weaken the structure of the stent, and this could cause inflammation, restenosis. And so to kind of avoid this, we would probably have to test if the metal ions are being released so that we could avoid inflammation or inflammation restenosis. And another problem that we could have is the sterilization if it's inadequate. And that would just lead to the contamination of the stent, leading to inflammation. And so we would have to do sterilization validation so that we could avoid this inflammation in the patient. And going on, we could also have problems with insufficient radial stent. And so this would make the shape of the stent abnormal and again lead to the restenosis thrombosis. So how we could maybe avoid this is do some radial force testing so that we could avoid the restenosis thrombosis. And the last issue that we thought about was stent migration. So it basically moves during the delivery. And so this could lead to the stent just being deformed, and that could cause vessel occlusion. And so what we would have to do is, again, the radial force testing and also stent retention testing as well.
Hi, this is Zeyu. Now I will move on to the no-clinical laboratory study plans for the novel scanning system. The no-clinical programs for this device consist of two main components. First, in vitro no-clinical bench testing, which focused on the engineering performance and the material safety. Second, in vivo no-clinical animal testing, which evaluates biological response in appropriate animal models. Both the stem and its delivery system, these two parts must satisfy or predetermined acceptance criteria for each test attributes. These criteria are established as part of the overall risk analysis. The primary purpose of bench testing is to ensure that the device meets all design specifications and design controls, and to provide a reasonable assurance of safety before the device proceeds into the clinical trials. Or in vitro bench testing method will be developed by the project teams and based on the FDA guidance listed on the slides, ensure that the test results are valid and acceptable for regulatory review. Next, because the device incorporates a novel aero material, so the material characterization is particularly important. These evaluations include a comprehensive assessment of the material's composition, shape memory, and the super elasticity of intravascular stents and corrosion resistance. These steps is critical to ensure that the material will not degrade or release harmful byproducts once implanted. Before we move to the next test, it's important to note that the stent designs generally falls in two categories: the balloon expandable and the self-expanding stents. So as a result, some detailed testing requirements differ depends on the final designs of the device we're talking about. So next, we conduct a stent dimensional and functional performance testing, which verifies that the stent's performance reliable during both the deployment and the long-term implantation. The testing includes the dimensional verification, the percent surface area for shorting, the recoil for the bottom expandable stents, the stent integrity, the radial stiffness, and the strength. They are especially important for the balloon expandable stents. The radial outward force is important for the self-expanding stents. And then the mechanical properties of both the raw material and the post-processing one. The strain and stress analysis the fatigue analyze and accelerate accelerate durability testing, the particular evaluation, the MR safety, and capabilities, radiopacity testing, because these case studies clarify the specifics. The device is for heart, for coronary stent system, so certain testing are not required. Yes, and next we move to the delivery system. It's also important is evaluated as a part of integrated stand system. So the test in focused on the dimensional verification, the delivery, the deployment, and the retraction performance for the balloon expandable stands. Additional testing, including the rated burst pressure, the FATO, the compliances, the inflation and the deflation time. Additional delivery system testing assists the culture bound strength, the TPO strength, the flexibility and kink resistance, the torque strength, and the coating integrity, the stem securements for unsheathed stems. Together, these tests ensure that the stems can be safely positioned without introducing additional procedure risk.
Okay, howdy y'all again. This is Grace. So to continue on this slide, we will focus specifically on shelf life testing. Shelf life testing is critical for the non-clinical in vitro bench testing program. Ultimately, its purpose is to evaluate how aging and the long-term storage can affect the safety and the performance of the NBMCSS prior to it being implemented in clinical use. So to do this, we need to repeat key bench tests on aged devices, which have been subjected to accelerated or real-time aging. And ultimately, this will help simulate conditions at the end of the labeled shelf life. So for the stent itself, we reassess dimensional verification, foreshortening, radial outward force, and particulate generation to confirm that the mechanical integrity and the deployment characteristics are maintained over time. We will also repeat delivery system testing, which will include dimensional verification, delivery, deployment, and retraction. And again, this will be to ensure that the system remains reliable after aging. Now, the balloon specific testing, such as the rated burst pressure, fatigue, compliance, inflation and deflation time, all of that will help confirm that the balloon performance is also not compromised with storage. Some additional catheter and system level tests, including bond strength, tip pull, flexibility and kink resistance, torque strength, coating integrity, and the stent securement, will also ensure that the delivery system remains robust and safe throughout its shelf life. But ultimately, it's also important to note that the radial outward force and the foreshortening are only repeated on the age stents in the device if it's self-expanding. And again, this just aligns and is consistent with FDA guidelines. Overall, these shelf life studies will help to provide assurance that the NBMCSS will perform as it was intended from the time that it is first manufactured to the time of its ending labeled expiration date. So now we're moving on to biocompatibility testing strategy. Ultimately, because the coronary stents are permanently implanted, they have direct contact with cardiovascular tissue and circulating blood for longer than 30 days. So this just means that they fall into the highest risk category for biological evaluation. So, as a result, we need a comprehensive battery of tests just to make sure that it's biologically safe and that the medical device, based on the contact type and duration. So, the different tests we will use will assess both the local and systemic biological responses, and this includes cytotoxicity, sensitization, and irritation, which again will just ensure that the materials are not causing any cellular damage or advice tissue reactions. Now, on the flip side, going more systemic wide, these will be evaluated through acute, subchronic, and chronic toxicity testing, while genotoxicity and carcinogenicity assessments will address the longer term risks that are associated with the permanent implantation of this device. Um, because this stent is in continuous contact with blood, we will also be ensuring that hemocompatibility testing is done. Again, this is just very paramount to evaluate the risks, may include thrombosis or other adverse coagulation responses. Furthermore, the implementation studies will work to assess the tissue response at the specific implantation site over time, which could include like inflammation and healing. Again, another important note to consider is that for devices containing nitinol, additional nickel ion release testing is going to be conducted just to evaluate the potential hypersensitivity or possible toxic effects related to that specific metal ion leaching. Again, next up is going to be the biocompatibility testing for the delivery system. This differs from the implanted stent just because the delivery system is externally communicating device. And so it has temporary contact, which is defined as less than 24 hours with the cardiovascular tissue and the circulating blood. So, due to the shorter duration of contact, the required biocompatibility testing battery is a little bit more limited than that for the permanent implant, but it's still critical to patient safety that we complete these. The testing is conducted in accordance with ISO 10993 Part 1, specifically using a risk based approach that reflects both the contact type and the duration. So these specific evaluations would include cytotoxicity, sensitization, and irritation testing, again, to assess whether the materials are causing any local tissue or cellular damage during contact. We will also be doing acute systemic toxicity testing, which will evaluate the potential for short term systemic effects following its initial exposure, while the material mediated prurigency assesses the risk of fever and other inflammatory responses that may be caused by the device materials. Because the delivery system contacts circulating blood, again, we will also be doing a hemocompatibility testing, just again to evaluate any risks of like thrombogenicity and other coagulation responses. Genotoxicity testing will also be included in this battery. It will be used to assess for the potential of DNA damage from any material exposure, even though that contact duration will be temporary. And ultimately, together, these studies will help demonstrate that the delivery system materials are biologically safe for their intended short term use, and then further support the progression toward in vivo testing and clinical investigation. So earlier I was talking about in vitro, and now we're going more into in vivo testing. So this slide is going to be examining the phases of non-clinical laboratory studies plan. Ultimately, the primary purpose of the in vivo testing is to gain a preliminary understanding of the device related risks. And it is also used to evaluate local and systemic biological responses, similar to what we had discussed in the previous slides, that otherwise wouldn't be able to be fully characterized through only in vitro bench testing alone, right? So, while the in vitro studies help confirm mechanical performance and material safety, these animal studies will actually allow us to observe how the NBMCSS interacts with a living system. And this will include interactions in contact with the tissue response, healing, any inflammation, and again, any potential adverse effects that may occur over time. For this process, the first clinical step is the selection of an appropriate animal model. This should be one that most closely replicates the human coronary anatomy. And this would also take into consideration the blood flow dynamics and other physiological responses. Ultimately, choosing the correct model is essential to ensure that the data are relevant and it's also transmittable to human use. If we look at the right-hand side, we see a decision tree, and it just kind of helps guide whether A, an animal test is necessary, but then B also helps determine what the most appropriate model and study design for the NBMCSS would be. And again, this would be based on prior bench testing, device risk, and existing scientific knowledge. So all the finalized animal studies will be conducted with full compliance with GLP, ensuring data integrity, ethical treatment of the animals, and regulatory acceptability of the results. Again, together, these in vivo studies will help provide critical evidence to support that the progression to first in human clinical investigations work.
Hi, that's Nikita again. So, before initiating human clinical trials, we will conduct GLP compliant animal studies using a porcine coronary model. Swine are the preferred model for the coronary stent evaluation because their coronary artery diameter and cardiac physiology closely match humans. We will evaluate both acute responses such as thrombosis and vessel injury and chronic responses, including neointimal hyperplasia and long-term device integrity. These endpoints directly address the key risk identified in our device evaluation strategy: thrombosis, restenosis, and structural failure. All animal studies will be conducted following 21 CFR Part 58 Good Laboratory Practice Regulations, and the protocol will be submitted to FDA in our pre-submission package.
This is Ying, and first overview of the regulatory background for clinical investigations of the NBMCSS. The object is to evaluate the safety and effectiveness of the NBMCSS for coronary artery disease. Next slide, please. Because it is a Class 3 medical device, the NBMCSS must follow the PMA pathway under 21 CFR 814. It means that the device is considered high risk and require both non-clinical and clinical evidence to support safety and effectiveness before market approval. Before any human study can begin, an IDE application under 21 CFR 812 is required. The IDE allows a significant risk device like a coronary stent to be legally tested in human while remaining under FDA oversight. Each study site must also obtain IRB approval, and every participant must provide informed consent under 21 CFR 50. This step ensures ethical protection and transparency for all subjects involved. Finally, the FDA monitors the entire process through IDE review and ongoing oversight, verifying that both data quality and participant safety are maintained. And how the NBMCSS trial is structured and where it fits in the overall development pathway. It usually starts with an early feasibility or first-in-human study, which focuses mainly on basic safety and device handling. The next step is the traditional feasibility study, where the near final device design is tested for preliminary safety and performance. Finally, it's the pivotal study, which is designed to generate definitive clinical evidence to support a PMA submission. And this is an example for a control trial comparing the NBMCSS to an approved commercial bare metal stent. It planned to enroll around 200 subjects, randomized 1-to-1 between the investigation and control arms. We measure whether or not a procedure is needed at the treated site. Follow-up will take place at 1, 6, and 12 months, and then annually for up to 3 years, allowing us to evaluate both early and long-term outcome. Oversight is provided by an independent data monitoring committee, ensuring patient safety and data integrity throughout the trial. Now that we have covered how the NBMCSS pivotal trial is designed, next is to make sure the study is conducted effectively and safely under the under GCP. Each clinical site must first receive IRB approval, confirming that the study meets ethical standards, and every participant gives written and informed consent after understanding the potential risk and benefits. Throughout the trial, adverse events are monitored and reported according to FDA and ISO requirements, and all personal data are kept secure and confidential. The sponsor oversees the entire study and ensure data integrity. Investigators are responsible for patient safety and proper protocol execution. Monitor checks data accuracy and compliance, and IRB or ethics committee continue to safeguard participant rights and welfare.
So as we conclude this presentation and class, we learned that the development strategy for the novel bare metal coronary stent system must follow a structured, risk-based approach that aligns with the FDA expectations for a Class 3 medical device. Through this project, we have established a strong foundation through quality system compliance and design controls, ensuring that safety and performance considerations were integrated throughout the development. Next, a comprehensive non-clinical testing program, including the in vitro bench testing, self-evaluation, biocompatibility testing, and the in vivo animal studies, was designed to systematically identify and mitigate device-related risks before human use. These non-clinical data support a phased clinical investigation strategy, first beginning with early feasibility and then finally progressing to a pivotal trial under the IDE while maintaining strict adherence to good clinical practice and human subject protection requirements. Ultimately, together, these integrated strategies should provide the scientific and regulatory evidence that is necessary to support a pre-market approval submission with the ultimate goal of delivering a safe, effective, and innovative coronary stent to patients. Thank you so much for your time. In a normal classroom, we would probably ask for questions, comments, and concerns, but that's not quite feasible here. But thank you again, and I hope you have a happy holidays and a joyous new year.